Hello everyone, and welcome to our webinar entitled Modulating the Gut Microbiome and the Role of Probiotics and Prebiotics. Our guest speaker today is Dr. Stephen Olmsted. My name is Michael Chapman, and I'm a medical education specialist at Genova's Asheville branch. I'm going to serve as the moderator for today's webinar. We would like to welcome Dr. Stephen Olmsted. Stephen Olmsted graduated from the University of New Mexico with distinction in biology and chemistry. He attended the University of New Mexico School of Medicine. He trained in internal medicine at Harvard Medical School, Massachusetts General Hospital. Dr. Olmsted completed a cardiology fellowship at the University of Washington, and he is board certified in both internal medicine and cardiovascular diseases. Dr. Olmsted served in the U.S. Public Health Service Commission Corps in the Indian Health Service, and for many years Dr. Olmsted was clinical assistant professor in medicine at the University of Washington Medical School in Seattle. He also served as advisor to the King County Natural Medicine Clinic during its inception and consulted to the Office of Alternative Medicine after it was first established at the National Institute of Health. He has long been an advocate of rigorous scientific research on complementary medical therapies, and he has more than 30 years of experience in clinical trials, registries, and basic research. In 2005, Dr. Olmsted joined ProThera as its Chief Science Officer, and Dr. Olmsted provides scientific support to both technical services and marketing at ProThera. He designs and directs clinical trials of ProThera products, including probiotics. Dr. Olmsted's current interests are in the development of innovative probiotic and prebiotic formulations and the use of nutraceuticals to disrupt dysbiotic biofilms. And one of the most common questions we get asked during the webinars is about availability of this presentation and the slide deck. These materials will be available on our website within a week of the webinar. If you're interested in having these resources, please click the Clinicians tab on the home page where you will find access to our webinars, or you can also log in to find the webinars in your MyGDX account. If you do not have a MyGDX account, please click on the Getting Started link on our home page. And without further ado, I'm going to turn the controls over to Dr. Olmsted.
Great. Thank you very much. As mentioned, my name is Stephen Olmsted, and I'm happy for this invitation to speak about one of my favorite topics, which is probiotics. I do work for ProThera. I'm their Chief Science Officer, and in the interest of full disclosure, we do market the Claire Laboratories hypoallergenic, high-potency brand of probiotics. So keep that in mind when you evaluate what I'm saying. So let's talk about the intestinal microbiota. So this is essentially an organ within us, and we'll get into this more. It's composed of over 100 trillion microorganisms. To put this in perspective, the microbiota, the gastrointestinal microbiota, contains 10 times the number of human cells. So although in terms of absolute mass it's quite small, in terms of numbers, it's quite large relative to us. So we are definitely composite organisms, meta-organisms, if you will. When you look at the amount of genetic information, the microbial genetic information just dwarfs the human genome. Most of what's going on genetically in our body is in fact microbial, and there's quite a bit of crosstalk that takes place. And we focused on the bacterial microbiota, but it's now being appreciated that there are other contributing organisms such as the archaea, certainly fungi, and to an extent that which we really have no idea, the millions of viruses, trillions of viruses that are within us. So all of this is taking part in forming the microbiota. We used to say that there was, you look in the literature, you'll see 400 species, 500 species, 600 species, but we now know there are thousands of species potentially, and we know this by sequencing sequencing the DNA of ribosomal RNA. And so we currently, and this was information that was published in Nature, we currently know that there are over 1,800 genera represented in the intestinal microbiota, and somewhere between 15,000 and 36,000 species can be detected. Nobody harbors all of these. And so the average person is going to harbor somewhere around 150 species. These are dominated by two divisions. Two phyla are very large organizational categories of organisms. So, for example, a phylum would be the Chordata. You know, so we're a member of the phylum Chordata, but obviously there's lots of diversity among members of the phylum Chordata, including the various different vertebrates, which is just one family in that. The microbiota is dominated by two divisions. The Firmicutes, these are gram-positive organisms, it's usually about 64% of the population, and the Bacteroidetes, these are gram-negative organisms, and that's usually about 23%, at least in Western populations. There's some variability in this, and we'll talk about that later. The vast majority of organisms in the intestinal microbiota are anaerobic, strictly anaerobic, fastidious. We can't culture most of them. That number ranges, I see in the literature, anywhere from 70% to 90%. A lot of these organisms, we just can't reproduce the conditions that they need for optimal growth, and it may well be that they can't be grown in isolation, that they need to be grown along with other organisms that perhaps are supplying them something critical that they need for optimal growth. I mentioned that you can conceptualize the microbiota as an organ, and I think that's a really important concept to grasp. So that this collection of microbes within our gut is in every sense an organ, like your liver or your kidney. It weighs about 1.5 kilograms. This is all the stuff in there. And the metabolic activity here rivals that of the liver. There is just so much that's going on in this community within our guts, primarily within the colon, the distal ileum and colon. We know that it's responsible for immune and GI maturation. We know that. That's been well established. Most of this has been established using what are called notobiotic animals, or basically germ-free animals, and then introducing known amounts of bacteria into their guts to see what happens. But the immune system and the gastrointestinal system don't mature normally. And they don't continue to function normally unless there is an intact, diversified, eubiotic microbiota. But we have been discovering that all sorts of other organ systems don't mature and develop normally, and this includes the central nervous system. And so, for example, normal myelinization in the brain doesn't occur unless there's a gut microbiota that takes place, and we don't understand fully the communications that go on that facilitate that. We do know there's a great deal of communication between the gut and the gut microbiota and the brain, and we know at least some of that is mediated via the vagus nerve. So people are beginning to understand what's going on, but we really don't have a full grasp. The gut microbiota is, of course, responsible for colonization resistance. If we don't have our normal commensal flora, we are going to be open to opportunistic and pathogenic infections. Throughout our lives, the gut microbiota modulates the immune system, and so it's the gastrointestinal tract that we come into contact with the external world. We don't think about it as the external world, but basically it is the external world. Everything from your mouth to your anus there is part of the external world. It's things that we take into our bodies, and this is where we are experiencing the world to a large degree. Think about that. And we now realize that the gut microbiota modulates metabolism. So it's very important in terms of glucose and insulin metabolism, weight metabolism, and there's a clear-cut role, although again we don't fully understand this, between the gut microbiota and dysbiosis and the risk of obesity and metabolic diseases like type 2 diabetes. The microbiota is really, it's so important, and it's not just this humans. This is an important aspect of biology in general, and in that all living creatures, all multicellular living creatures, are living in symbiosis with some microbial organisms, and we are living in symbiosis with a lot of them, and it's important for our health that we be exposed to human microorganisms, and we get these normally from our mothers. And so there's a wonderful system that's developed whereby a mother can transfer her own microbiota from her gut into her breast milk, and the infant receives this while nursing. And so this is mediated by dendritic cells, so immune cells within the gut, which are actually able, as you can see, these little dendritic cells here in the middle bottom picture with these little tentacles. And so they send these out, and they can actually go straight into the lumen of the gut, sample the gut microorganisms, sample gut bacteria, bring them back in, not harm them, transfer them to lymphocytes, travel via the lymphatic system, are released in the breast and into milk where the infant consumes them. So this is some data on real-time polymerase chain reaction testing of breast milk. So 43 nursing mothers, and you can see that on average, there's somewhere between 200 and 1,000 CFU per milliliter of breast milk, and some women have none, and I would suggest that that's probably not normal. I would wonder about whether those mothers actually having a dysbiotic microbiota. But you can see that it's quite normal for women to pass bifidobacterium onto their infants while nursing. Other organisms are passed on to their infants, including clear-cut probiotic organisms like Lactobacillus gasseri. Also, some streptococcal species are passed through into the breast milk, and staphylococcal species, Staphylococcus epidermidis, for example. So a number of different organisms are passed on to the infant, and that's part of how infants acquire a normal eubiotic microbiota. But we now know, I mean, a classical teaching, if you look back just a few years ago, the dogma has been for 150 years that the fetus is sterile unless there's something wrong, that amniotic fluid is sterile, that the placenta is sterile, and that acquisition of the gut microbiota only begins essentially during birth and then following birth, and that's where we get it. But this turns out to be, as so many other things that I learned over my career years ago, this turns out to not be true. And we now know that amniotic fluid is not normally sterile, that you can find small populations of bacteria, and all of these different genera are listed here on the right of the slide. As you look at it, you can find Enterococcus, Staphylococcus, Streptococcus, Klebsiella, Bacteroides, Bifidobacterium. All of these can be found in the amniotic fluid. And then, of course, because the fetus is drinking the amniotic fluid during development, these then inoculate the meconium. We also know that the placenta now harbors its own microbiome that is involved with protecting the fetuses, and if there's something wrong with that, maybe associated, if there's a dysbiosis of the placental microbiota, this is associated with preterm deliveries, especially preterm deliveries before 28 weeks. So the microbiota occurs before birth, starts to occur before birth, but really takes off after birth. So the baby, as it's being born, is covered with the microbiota of the cervix, of the vagina, and of the perineum. And then there's great homology between the organisms you can find. For example, E. coli serotypes in the nasopharynx of an infant after birth. That's going to match up perfectly with E. coli and found in the mother's feces, for example. A lot of these Streptococcus and Enterobacteriaceae organisms are aerophilic, and they get into the intestines. They're swallowed by the baby, and they grow, and they essentially set up the anaerobic environment that's required for Bifidobacterium and Bacteroides and various Clostridia clusters. And so you get an establishment of this predominantly anaerobic flora that occurs, within the first week of life. And then if the baby is breastfed, the baby develops a Bifidobacterium predominant microbiota, and that's normal. And babies who are not breastfed don't develop a normal Bifidobacteria predominant microbiota, and that has health consequences for them. And babies that are born by cesarean section don't develop they have a dysbiotic microbiota because they haven't had that initial exposure. And of course, the worst thing is to be born by C-section and be formula fed and to spend a lot of time in the hospital, and those poor infants don't develop anything close to a normal microbiota. But there is this microbiota that develops during infancy, then becomes more adult-like after weaning and exposure to solid foods. And you have a predominance of the firmicutes, the gram-positives, which include things like Lactobacillus, Clostridium, Bifidobacterium. I'm sorry, not Bifidobacterium, but Lactobacillus and Clostridium, and Eubacterium. And then you have the Bacteroides, and these become more predominant as one is older, and you have a decrease in Bifidobacterium as one ages. And there are many people that speculate that this decline in Bifidobacterium numbers with age contributes to immune dysfunction in the elderly. If all goes well, you come up with a balanced eubiotic microecosystem that keeps potentially harmful bacteria in check, like Pseudomonas or Proteus or Staph, and has good numbers of potentially helpful bacteria, which of course are modulating your immune function, are aiding in the digestion and absorption of nutrients, stimulating gut motility, synthesizing vitamins, and inhibiting pathogens and potential pathogens. It's a complicated place. This is a complicated slide, but it's just to really illustrate two things. One is that everybody's microbiota is a little different. So we may have similar organisms, but the balance is going to be different from person to person. So for example, here's a healthy 28-year-old woman on the upper left, and she's got over 20% Bifidobacteriales. So she's got a really healthy Bifidobacteria population, and that's a good thing. And then here's this 37-year-old man, and his bifidobacterial population is down around 5%. And so he's got much more in terms of Clostridia, and you know, there could be some potential health implications for that. So all of these, we just need to realize that there's a lot going on, and that when we're trying to modulate this, we're doing this with tools that aren't very precise or fine. There's been interest in microbiota enterotypes, so that the initial studies published a few years ago in Nature suggested that people could be broken out into those who had Blautia predominant, or Prevotella predominant, or Bacteroides predominant populations. This was based on largely Western populations, as you can see, with some Japanese people studied. So there's a question about, gee, are you going to extrapolate this to the whole world? That there might be some differences, but some additional larger studies have tended to suggest that healthy people do break down into different enterotypes. Probably a lot of this is based on diet, but everybody's not the same, and so the approach to dysbiosis isn't going to necessarily be the same in everybody. Just keep that in mind. The gut microbiota, one of the most important things that it does is to maintain intestinal integrity. And specifically, when we're talking about intestinal integrity, one of the things that encompasses is healthy barrier function. So you don't have a leaky gut, you have good gut barrier function. The integrity of the intestinal barrier is maintained intact, so you're only absorbing things that you need to absorb, and other things like toxins or pathogens are not coming into your body. The commensal bacteria helps with this because it stimulates mucus production. Mucus is obviously an important protective barrier. We know, for example, that people with inflammatory bowel disease have less thick mucus than people with healthy guts. The commensal bacteria stimulate the enterocytes, or the cells lining the intestine, to secrete defensins. These are molecules that, as the name suggests, defend the body against pathogens. They have toxic effects on pathogens. The commensal bacteria produce microcins or bacteriocins. You'll see it referred to. These are molecules with toxic effects on pathogens, and part of the way that the commensal bacteria keeps pathogens in check, keeps them from translating across the mucosal barrier, the gut barrier, into the submucosal areas and regional lymph nodes. And one of the most important things that the commensal bacteria do is modulate immune function, and they do this primarily by increasing activity of regulatory T cells. And here's an illustration of this. People used to talk about how you had naive T cells. These would be the Th0 cells, and then these T cells could differentiate into helper 1 T cells, Th1 T cells, and these were responsible for cell-mediated immunity, or they could differentiate into helper T cell 2s or Th2 T cells, and these were associated with humoral immunity, and overexpression of Th1 cells was with tumor necrosis factor and interferon gamma, and interleukin-2 was associated with autoimmune diseases if it was overexpressed. While overexpression of Th2 T cells with various interleukins 4, 5, and 10 was associated with allergy. So people used to talk about a Th1 to Th2 ratio, and you need to, if you had too much Th1, you need to balance it by increasing Th2, and that turns out to be completely wrong. And the important ratio is between regulatory T cells, the Tregs, and the effector T cells, so all these other T cells. And in addition to Th1 and Th2, there are also other helper T cells, Th17s, associated with cellular immunity, and so is Th22, just as Th9 is associated with asthma. And it turns out that these cells are not even terminally differentiated. So a Th2 cell can turn into a Th1 cell, or a T regulatory cell that should normally damp down excessive immune responses can turn into a Th1 or Th17 cell. What's important is that it are robust populations of T regulatory cells, and that's where the gut microbiota and that's where probiotics play a major role in regulating and modulating immune function. So, how do we modulate the gut microbiome? We've got 5 different modalities, general big modalities that we can use to modulate the gut biome, and I'm only going to talk on about 2 of them, but I want you to know about all of them. One is diet, and this is probably much neglected, and is something, you know, I could spend another hour, a couple hours on is talking about diet, but we shouldn't forget about diet, and it almost strikes me as pointless to take probiotics or take inulin fiber and have a diet of junk food. I mean, it's like, what are you trying to do? So if you don't address the diet, and if you don't clean up the diet, and if you don't encourage the intake of a lot of soluble, for us, undigestible fiber and fresh fruits and vegetables, and legumes, then it's going to be hard to really make an impact with other modalities like probiotics and prebiotics. You can also modulate the gut flora with antibiotics, and so, for example, what you'll immediately appreciate, for example, antibiotic therapy, rifaximin of small intestinal bacterial overgrowth. What are we doing? We're trying to modulate the gut microbiota. We're trying to decrease populations. There's a whole literature on antibiotics and Crohn's disease. So you know, it's the problem with antibiotics is that how do we know that we're just killing things, the bad organisms, the organisms that are responsible for diseases, and you either have a drug with really limited spectrum like rifaximin that doesn't appear to harm healthy commensal bacteria, or you have other antibiotics that are potentially more toxic. But this is a way of modulating the microbiota. And then finally you have fecal transplantation, again way beyond the scope of this presentation, but a very exciting area. Obviously what's available to us as probiotics is really limited in terms of the actual populations composing a healthy gut. Fecal transplantation allows you to transplant what you hope, you do your best to document, is a healthy microbiota from a donor into somebody significant problem like recurrent Clostridium difficile disease, which is obviously potentially fatal disease, or it's even being done in inflammatory bowel diseases with some success. And in this country anyway, the FDA says that it's regulating this and it's experimental, but it's very exciting. So in terms of definitions, probiotics, the term was first used in 1965, and what it referred to was microbially produced, so growth factors produced by microbes that promote good microbes, basically. And so Fuller in 1989 expanded that to say that these were live microbial feed supplements. So we should all know that really where probiotics are extensively used and have great impact, especially as an alternative to antibiotics, is in animal husbandry and is in raising chickens and pigs. And so there's a lot of work that goes on in that, and that's where probiotics as live microbial feed supplements that benefited the host, that's where the concept was really developed, and then it was sort of ported over to humans. And a currently accepted definition of probiotics is these are viable microbial food supplements that have a beneficial influence on human health. So so a couple things. It has to be microbes. They have to be viable. They can't be dead if it's going to be a probiotic. And they have to have a health benefit, sort of by definition. If there's no health benefit, it's not a probiotic. Now, Prebiotics are non-digestible substances, so substances that we don't have the enzymes to digest that stimulate the growth and or activity of selected GI microbes. So it doesn't mean that you have to increase populations of certain beneficial microbes. You just have to increase their activity. And in fact, there are many people, there's this concept of functional dysbiosis. So there may not be something on a stool test that really that jumps out at you in terms of too much of this or too little of that, but functionally in terms of what sorts of enzyme activities and what sorts of nutrients are being produced, that might be where the dysbiosis is, and so that could be moderated by prebiotics, and then a symbiotic is just a combination of probiotics and prebiotics. That's all a symbiotic is. So just a brief history of probiotics. I like the concept of essential probiotics, like essential vitamins. We have evolved with these organisms, and in fact, there's a great paper in Nature this past month looking at humans and their nearest relatives, chimpanzees and gorillas, and we have evolved with similar firmicutes and similar Bacteroidetes and other organisms. However, in terms of species differentiation, we've all had our own microbiota as humans different from other primates for 15 million years. And we get them from our environment, and we get them from our mothers to begin with, and then we get continual exposure to them as we take in probiotic organisms from our diet, and we find probiotic organisms naturally on fruits and vegetables, and I'm sure that humans discovered thousands and thousands of years ago natural fermentation of different fruits and vegetables and appreciated them. And we know that 10,000 years ago that people started fermenting beverages on purpose, and in fact there's one whole theory about the origin of agriculture, that it had nothing to do with assuring a good supply of food, a reliable supply of food, and had everything to do with securing a reliable supply of beer. We know that yogurt was developed about 3,000 years ago. Basically, fermented foods have been part of the human fabric for a long time. And Hippocrates, of course, the father of medicine, was prescribed sour milk. He realized that there was therapeutic benefit in giving patients with GI disorders sour milk. Pliny the Elder also expounded on the benefits of sour milk, and in fact, Francis I of François Premier was cured. At the time was allied with the Sultan of Turkey, and your complicated European politics, and was sick, gastroenteritis, and was cured by the Turkish physician, cured of dysentery by yogurt. But the godfather of probiotics is Eli Metchnikoff, who won the Nobel Prize in 1908, not for probiotics, but for the description of phagocytosis. But he was the first person to really he worked at the Pasteur Institute, and he was quite aware of microbes, and he was the one that made it, that understood that taking in fermented foods like yogurt made possible the prolongation of a healthy life, and he wrote this in his seminal work, The Prolongation of Life, which was published just about 110 years ago. So the types of probiotics, most of the probiotics that are available to us are organisms that have been used in food and beverage production. So keep that in mind. That's why we have these. That's why the FDA lets these in on the market, is that there has been a lot of experience, in many cases thousands of years of experience with these organisms. So they're generally divided into lactic acid bacteria, lactobacillus species. Of course, some people would quibble about whether the bifidobacterium species should, strictly speaking, be called a lactic acid bacteria. Acetate's a more preferred end product for them, but many people lump them in. Streptococcus thermophilus, this is a traditional yogurt starter, highly adapted streptococcus, not pathogenic. Enterococcus faecium, lactococcus, leuconostoc, pediococcus. These are all organisms that produce large amounts of lactic acid. Then there are the non-lactic acid bacteria. There's the bacillus species. These are often referred to as soil-based organisms. There's the Propionibacterium species, and there's E. coli nisla. It's a specific strain of E. coli isolated from a German soldier in 1917. Everyone else in his unit died of dysentery. He survived, and the German physician astutely noted that it was probably because of his microbiota and isolated this particular E. coli. This is available, and there's some good evidence that it's beneficial in Crohn's disease, for example. And then the non-pathogenic yeast, Saccharomyces boulardii. Lactobacillus characteristics, these are gram-positive rods. They can also have a coccobacillary form. They don't make spores. Broadly divided into homofermentative, so the 85% plus of their fermentation end product is lactic acid, or they're heterofermentative, so in addition to lactic acid, they also make carbon dioxide, ethanol, and acetic acid. They digest and metabolize proteins and carbohydrates, make B vitamins, vitamin K. They break down bile salts. They are intimately involved with modulating immune function, and they inhibit a variety of pro-inflammatory mediators. And clinical studies and animal studies over the years have shown that lactobacilli have active antimicrobial properties against a wide variety of pathogens, gram-positives, gram-negatives, and yeast. This is a list of probiotic lactobacilli that are generally available. I'm only going to discuss a few of them in detail. Lactobacillus acidophilus, this is the prototypic probiotic. If anybody's ever heard of a probiotic, what they've heard of is Lactobacillus acidophilus. Important to realize that a lot of strains that 20 years ago were classified as Lactobacillus acidophilus have since been reclassified, so that can be a little confusing when you look at the literature. Currently, it's thought that Lactobacillus acidophilus is a transient species, meaning that it's only going to be there if you're taking it in your diet. A lot of what used to be classified as endogenous Lactobacillus acidophilus has now been reclassified as like Lactobacillus salivarius or Lactobacillus gasseri. Lactobacillus is an obligate homofermentative organism. It never switches to heterofermentative. It has to make a lot of lactate, and it makes both isomers of lactic acid. It's very hardy, resistant to acid, bile, pepsin, pancreatin. It has over 20 known peptidases, and it has been shown to break down both casein and gluten, which are problematic polypeptides. Ferments lactose, and in fact there's a literature on Lactobacillus acidophilus for lactose intolerance, and it can reduce gut concentrations of various carcinogenic enzymes, although the studies have yet to be done that show that probiotics can reduce the risk of colon cancer. Lactobacillus rhamnosus, this is probably the most studied probiotic. It's relatively fragile. It's much more susceptible to stomach acid. It's generally transient, although people can isolate it in the gut. It's a heterofermentative, and it only makes the L-positive lactate isomer. It's got great colonic mucosal adherence, although that's done in vitro, and how relevant that is to in vivo, we don't know. It makes more peptidases than any other lactobacillus, and it enhances innate and acquired immunity. That antagonizes various pathogens like rotavirus, C. difficile, and E. coli. Lactobacillus casei is another adaptive probiotic closely related to rhamnosus, again, transient species. And one of the things that this has is a large number of proline-specific peptidases, and these are particularly useful in breaking down casein and gluten, because these are polypeptides that have a lot of proline in them. It also makes the L-positive and only the L-positive form of lactic acid. And although it's transient, it's interesting in that its presence is required for differentiation, for normal differentiation of dendritic cells. So it's amazing. So you require this transient species for normal immune maturation. Lactobacillus plantarum. This is ubiquitous on plants. That's why there's a picture of vegetables here. Generally lacking in modern diets. So if you do stool cultures from people in Africa who are eating a traditional diet with a lot of fiber and legumes, they all have Lactobacillus plantarum. And humans have modern Western diet. People consuming modern Western diets have like 25% of them have Lactobacillus plantarum if they're not taking it as a probiotic. Very hardy organism, resistant to bile and acid. It facilitates induction of IL-10. This is really important for T-cell regulatory function and decreasing both allergies and inflammation. And it strongly supports intestinal barrier function and reduces translocation of gut flora. Now, Bifidobacterium. These are the second major class of probiotics, gram-positive. They don't make rods. They often have a Y shape to them, especially when they're stressed. That's why that's called Bifidobacterium. They're strictly anaerobic and fastidious. The one exception there is Bifidobacterium lactis. They have a unique metabolic hexose pathway called the bifidus shunt. They all break down lactose. They ferment a variety of non-digestible oligosaccharides. And they synthesize B vitamins, vitamin K. Again, they're intimately involved with modulation of both innate and acquired immunity, and inhibit a variety of pro-inflammatory mediators, and inhibit a variety of different pathogens. And when you look at different disorders and disease states, what you will see over and over again is that disease or ill health is associated with reduced bifidobacterium populations, and robust bifidobacterium populations are associated with gut health and immune health. These are the major bifidobacterium probiotics, and we'll discuss some of them in a little more detail. So Bifidobacterium longum, this is often, so if you're fortunate enough to have a dominant Bifidobacterium microbiota, it's usually going to be the Bifidobacterium longum that's dominant. It ferments a variety of oligosaccharides, like they all do, resistant to bile salts. It's able to inhibit enterotoxigenic E. coli receptor binding, inhibits Clostridium proliferation, enhances GI secretory IgA response to dietary antigens, hence it reduces dietary allergies, and it modulates cytokine responses to respiratory antigens. And you might think, oh, well, how could it do that? But keep in mind the whole concept of the common mucosal immune system, so that if you have something going on in the gut, associated lymphatic tissue, that's going to affect immune tissues elsewhere in the body. Bifidobacterium bifidum there are smaller populations of this organism, this healthful organism, found in allergic infants. Populations of bifidum, B. bifidum, tend to decline with age. Talked about how people speculate that reduced immune function, increased susceptibility to disease in the elderly may be in part related to reduced bifidobacterium populations. Obviously, with its ability to suppress total and antigen-specific IgE production, it's going to be protective against allergies. It enhances IgA response to Clostridium difficile, antigen toxin A rather, And it's been shown together with Lactobacillus acidophilus to support a normal microbiota during antibiotic therapy and reduces testing for C. difficile toxin A. Bifidobacterium infantis, as you can deduce from the name, this is very frequent in infants, rare in adults. If you're a breastfed baby, your microbiota should have lots of Bifidobacterium infantis. That's a good thing. It suppresses populations of Bacteroides vulgaris, reduces pro-inflammatory cytokine production, improves various cytokine ratios in irritable bowel syndrome, and together with acidophilus, L. acidophilus, reduces the risk of necrotizing enterocolitis in very low birth weight infants. So, B. breve, Bifidobacterium breve, this has been shown to reduce the populations of pathogens, Bacteroides flagellus and Clostridium perfringens. It improves weight gain in very low birth weight infants, stimulates Peyer's patch B-cell proliferation. So again, it stimulates normal maturation of the gastrointestinal immune system. Relatively hardy organism, resistant to acid, bile, pepsin, pancreatin, and enhances B-cell antibody production. In fact, there's some work that it improves the response to immunizations. It can antagonize rotavirus and decreases rotavirus shedding. Just going to touch briefly on bacillus characteristics. These are frequently misbranded, misidentified organisms. So if you're going to use bacillus probiotics, be sure that it's from a reputable manufacturer. These are sporulating rods. These are facultative anaerobes. People used to think that any bacillus that was in the gut was just there because it was on the carrot that you consumed or the potato or whatever, and that these were transient. But it turns out that with some species like Bacillus subtilis, there are more of them in the gut than you could explain just simply by ingestion. So low levels of them are definitely commensal. And bacilli are commonly used in fermented fish and manioc and soy food. So, for example, that Japanese food natto, that's produced by Bacillus subtilis. So there's evidence that they're useful in irritable bowel syndrome and prevention of antibiotic-associated diarrhea. They can inhibit pathogens. They make bacteriocins and lipopeptides, and they definitely promote colonocyte health. So there's a role for them, but they're frequently misbranded. So make sure you're getting them from a reputable company. These are some of the Bacillus probiotics that are used. Primarily Bacillus coagulans and Bacillus subtilis are the most important. Now, Strep thermophilus is a lactic acid bacteria. This is a traditional cheese and yogurt starter. It's highly adapted to living on lactose. It's transient. There's protocooperation with Lactobacillus bulgaricus, which is another yogurt starter. And it has a variety of different clinical uses, from rotavirus to improvement in ulcerative colitis and maintaining remission in ulcerative colitis and other inflammatory bowel diseases. And there's some evidence that it can provide protection from pathogenic Streptococcus species. Saccharomyces boulardii, this is this probiotic yeast, transient, non-pathogenic. It was discovered by a French mycologist, Boulard, who found it in Southeast Asia. People were brewing a tea from lychee nuts to treat dysentery, and he figured out that it was the Saccharomyces boulardii that was the active principle in these teas. This is a very hardy organism and does an amazing number of things. Increases brush border enzyme activity. It's the one intervention that is going to increase intestinal short-chain fatty acids and secretory IgA concentrations more than anything else that you can do. It has a powerful protease that inhibits Clostridium difficile toxins A and B, antagonizes Candida in the gut, and it's the one probiotic that has been shown to be effective, at least as an adjunct against unicellular parasites like Entamoeba histolytica and Giardia. And there's actually one study that showed that it was just as good as metronidazole in treating Blastocystis hominis. Now, probiotics, the documented uses of probiotics, there's a literature that supports the use. There are meta-analyses that supports the use. It's not really controversial. People can argue about the quality of the studies and that sort of thing, but there is a literature that supports the use in antibiotic-associated diarrhea, Clostridium-associated diarrhea, various other diarrheas, community, travelers, a use in inflammatory bowel disease, irritable bowel syndrome, and vaginal dysbiosis, so Candida vaginitis and bacterial vaginosis, recurrent urinary tract infections in women, allergies, atopic dermatitis, eczema, lactose intolerance, hyperlipidemia. There are things that people are looking at now, like probiotics in weight loss, probiotics in blood sugar control, probiotics in depression, probiotics in anxiety. So there's a lot of the documented uses are expanding for probiotics. Well, let's just look at some of these. The antibiotic-associated diarrhea, this is disrupted GI microbiota due to antibiotics. That's what this is due to. And so everybody with antibiotic-associated diarrhea, the common denominator here is that they have decreased colonic concentrations of short-chain fatty acids. But up to 50% of the cases are due to Clostridium difficile, but everybody's got decreased short-chain fatty acids. So basically the antibiotics are killing off the organisms that make short-chain fatty acids, especially butyrate, so acetate, propionate, and butyrate. And butyrate is, in particular, is important. It's nutritive to colonocytes. Colonocytes don't get this. They don't do what colonocytes do, which is absorb water, absorb salt. And so you get diarrhea. It's common, how common it is, it depends on the series you look at, but it's common. So, children anywhere from 11 to 50%, adults 5 to 39%, take your pick, depends on the series, patient population, etc. And the definition is that of antibiotic-associated diarrhea is any diarrhea otherwise unexplained that takes place within 2 months after a course of antibiotics. Probiotics work. And so here's a large meta-analysis that was done by Lynn McFarlane, and she looked at all of these different trials, and these are different probiotic preparations and formulas. So, for example, SB is Saccharomyces boulardii, LGG is Lactobacillus rhamnosus GG, Bacillus coagulans, Bifidobacterium lactis. Enterococcus faecium. So all of these different Lactobacillus acidophilus. So all these different protocols. And what Lynn found was that out of 25 randomized clinical trials, that the overall risk reduction was 0.43. So that's a 57% reduction in the risk of antibiotic-associated diarrhea. Really fairly profound. And probiotics should definitely be used when you're prescribing antibiotics. I just don't know why a clinician wouldn't do that. So in terms of what should be used, what the literature supports is Saccharomyces boulardii. So then the dose should be 10 billion CFU a day. And optimally, that should be combined with Lactobacillus rhamnosus, so 5 billion CFU a day. And I think it would be very advisable to use a multi-strain formula that also contains some bifidobacterium, because we saw some of these species antagonize Clostridium difficile and decrease its proliferation, that sort of thing. Also, one should consider using prebiotics, specifically inulin prebiotics, to increase short-chain fatty acid producing population. So we don't have Eubacterium and Roseburia available to us as probiotics, but we can increase their populations and activity by giving people prebiotics. In general, I think you should use probiotics at the same time, concomitantly with the antibiotics, in order to minimize the effect of the antibiotic on the probiotic and to maximize survival of the probiotics that you take in. I suggest people take their probiotics an hour before or 2 hours after the antibiotic dose, and I've always suggested a 30-day total course of probiotics. So even though your antibiotic treatment may be a week or 10 days, I've always suggested a 30-day total treatment. Now, inflammatory bowel disease, so this includes Crohn's disease, ulcerative colitis, and pouchitis. There are differences in these. Pouchitis is disease that's involving a portion of the ileum that's being used as a rectal pouch following surgery for, in almost all cases, ulcerative colitis. And we know that inflammatory bowel disease is due to an abnormal response to certain commensal bacteria. So that if you have no bacteria, or let's say, for example, you have rectal ulcerative colitis and the surgeon does an ileostomy to divert the flow of intestinal contents, you can treat the disease by decreasing exposure to intestinal contents and commensal bacteria. People with inflammatory bowel disease have high numbers of Bacteroides, Enterobacteriaceae, and Peptostreptococcus, and, you guessed it, low numbers of Bifidobacterium. Really, I have to say that, so the animal models, just, it seems like probiotics should just be gangbusters. So here you have this rat model of inflammatory bowel disease, this HLA-B27 transgenic rat, and if you raise it germ-free, so there's no intestinal microbiota, the rat does not get colitis. But when you introduce cecal bacteria into the rat, you get a very, very aggressive colitis. So there's this interaction with this genetic predisposition and the presence of cecal bacteria. If you introduce, remember, these are gnotobiotic organisms, so you have control over what you are introducing. You know what is in the gut. So if you put in just Bacteroides vulgaris, you get colitis. So that's one of the things that's in the cecal mix that's giving you colitis. But it's not as bad as everything that's in there, so it's just one thing. If you introduce E. coli, so this aerobic Enterobacteriaceae, there's no colitis. So we know that that's not the problem. If you introduce Lactobacillus rhamnosus plus cecal bacteria, you completely protect this rat from getting colitis. So probiotics should just be wonderful, but they've been disappointing. And so here are studies that are looking at probiotics in Crohn's disease and ulcerative colitis and in pouchitis. And with the exception of pouchitis, for which gastroenterologists say that the evidence is good to use probiotics, most of these studies are negative. What about that? Well, some of this is study design. So for example, if we look at this study, so here's treatment of Crohn's disease using Espulardi. So you're using Espulardi as an adjunct. So there's standard therapy, mesalamine 3 grams a day versus mesalamine 2 grams a day in Espulardi. And the number of clinical relapses was significantly less. So is the appropriate endpoint being studied? So probiotics probably don't have a big role in treating acute flares, but they clearly, I think, have a role in keeping people in remission. You know, here's a study of people in Crohn's disease in remission, and so this is involving a preparation called VSL number 3, which is simply a multi-species high-potency probiotic based around Streptococcus thermophilus. And here you see that this is a study that shows probiotics don't work, right? But there's only 40 patients. There was a 50% reduction in relapses, but the numbers were not significant. So is this 50% reduction not real? It's just by chance, or was it not significant because this is an underpowered study? Probably an underpowered study. That's the problem with probiotic studies, is they often have too small of a sample size, and that might be why probiotics in the literature have turned out to be disappointing in inflammatory bowel disease. They're not disappointing in the treatment of pouchitis, and again, most of the data comes from VSL number 3, this high-potency, multi-species probiotic, and it's clearly effective. Now, one of the things about probiotics, it's become clear that single probiotic species don't work in inflammatory bowel disease. They just don't work. But if you but combining them with inulin, so for example, prebiotics, so for example, in this study here at the bottom where they combined inulin and fructooligosaccharides with Bifidobacterium longum, they had great results. 18 patients, active ulcerative colitis, and they were assessed clinically, sigmoidoscopically, and histologically, and they had significant improvement clinically, and improvement in their sigmoidoscopy scores, and decreased tissue inflammation, increased healing. So the combination of Bifidobacterium probiotic with prebiotics is beneficial in this population. People have looked in an open-label study at inulin alone, 15 grams a day. Don't start anybody at 15 grams a day, but work up to that. And again, significant improvement and also increased Bifidobacterium populations. Prebiotics are a great way of increasing Bifidobacterium populations. And again, inulin has been used alone in pouchitis, again with some benefit. So probiotics plus prebiotics are the way to go in inflammatory bowel disease. So my suggestions for inflammatory bowel disease, for remission, use Saccharomyces boulardii. I tend to advise people if they get a response to go a month without it, 3 months on, a month without. Just because Saccharomyces boulardii isn't part of the normal microbiota. For Crohn's disease, I would combine that with inulin-based prebiotics and a multi-species high-potency probiotic in the dose range of 200 to 400 billion CFU a day. For ulcerative colitis, the dosing, same dose for Saccharomyces boulardii, higher doses for the multi-species probiotics for in the range of 400 to 800 billion CFU. For pouchitis, multi-species probiotics and inulin-based prebiotics are the way to go. Irritable bowel syndrome. This is a common problem, 10 to 20% of adults. Tends to be a benign disorder, benign in that, not that it causes people a lot of misery, because it does, but benign in that you don't die from it. It's characterized by abdominal pain, bloating, diarrhea, constipation. There's problems with intestinal motility. There's increased visceral sensitivity, and we know that people with irritable bowel syndrome have GI dysbiosis. They have decreased lactobacillus and Conicella populations and increased Clostridium, Ruminococcus, and Veillonella populations. They have this increased hypersensitivity. One of the things that we now know is that probiotic organisms can modulate production or perception of gut pain, and this is a study from Nature Medicine 2007 where they looked at Lactobacillus acidophilus, and basically what they found was that Lactobacillus acidophilus increased numbers of opioid and cannabinoid receptors in this mouse model and was as effective at relieving pain as morphine. So modulation of pain perception may be part of why probiotics are beneficial in irritable bowel syndrome. In a meta-analysis of 19 randomized controlled trials, probiotics were significantly better than placebo, and the risk reduction was 0.71, so there was a 29% improvement. The number needed to treat was small, it was 4, but do keep in mind they're not going to work for everybody, and you're going to have to treat 4 people before you see 1 improvement. The organisms that are found to be efficacious, Bifidobacterium infantis, Lactobacillus plantarum, and again, high-dose multi-species probiotics.
Hi, Dr. Olmsted.
So what I generally recommend, sorry?
Sorry to interrupt you. I just wanted to jump in real quick because we just have a couple minutes left.
Right.
And we have a hard out at the top of the hour, so I was just going to give you a chance to sort of wrap up and then maybe take over controls do a wrap-up.
Okay. So basically with irritable bowel syndrome, what I recommend is 200 to 400 billion CFU a day. Consider inulin, although I'm doing that less and less. If there's a response, people need to take it indefinitely. You need to rule out gluten sensitivity, food allergies. Consider digestive enzymes. Just with allergies, dermatitis, this is a pandemic. It's affecting a lot of people, and altered gut microbiota is implicated. We know that a number of different probiotic organisms induce immune tolerance, can be beneficial. When people have done meta-analyses, we found that probiotics are important and effective in reducing the incidence and severity of allergies. They're effective when given to pregnant women. They're effective when given to breastfeeding women to reduce allergies in the infants. And they have been shown to reduce atopic dermatitis and eczema during 7 years of follow-up. Probiotics also reduce symptoms and improve quality of life measures in allergic rhinitis. Probiotic selection, you face between monostrain, multi-strain, multi-species probiotics. The evidence really favors multi-species probiotics. Although you will constantly hear it said that that benefits are strain specific. This is a marketing claim, and there's very little science that backs that up, even though academics and regulators are parroting this. It would require head-to-head studies, and these are rare. And when you look at the reason for this, there's not very much difference between strains. And so this is gene sequencing in a patented strain, Lactobacillus acidophilus NCFM and Lactobacillus acidophilus LA14, which we use in our probiotics, and they are 99.98% identical. And the differences that are there are of no importance. Start low at like 10 to 25 billion a day and increase slowly. This minimizes symptoms of gas and abdominal discomfort and diarrhea. I suggest taking probiotics with food. The reason for that is that the pH of the stomach with food is significantly higher. Probiotic survival will be better than if you take it on an empty stomach. Probiotics are safe. Doses actually greater than 3.6 trillion have been used in adults, and 110 billion CFU per kilogram in infants have been used safely. They've been used in immunosuppressed patients, and they've been used in hospitalized patients. So in conclusion, multi-species probiotics are supported for use in a variety of different GI conditions. Saccharomyces boulardii supported for the use in antibiotic-associated diarrhea, Clostridium-associated diarrhea, irritable bowel or inflammatory bowel disease, and traveler's diarrhea. Probiotics are useful for the prevention and treatment of allergic disorders. Prebiotics also show benefit for inflammatory bowel disease and allergies. Clinical trials, well-designed, well-conducted clinical trials with relevant formulas are desperately needed in the area, as well as outcome studies. Specifically, I am interested in aging and cancer prevention. So I'd like to thank you for your attention, and open up the floor to questions.
And unfortunately, we don't have time for question and answer at this particular point, just in the interest of time. And I want to thank you, Dr. Olmsted, for a terrific presentation. Sorry I had to jump in there. If you have any additional questions, you can contact us at these phone numbers listed on this particular screen, or by calling customer service. And lastly, just wanted to encourage you to look for upcoming webinars on www.gdx.net. Next month we have Dr. Kathy O'Neill-Smith who will be presenting on using the elimination diet in clinical practice. And thank you one more time, can't thank you enough, Dr. Olmsted, for a great presentation.
Great, thank you, you're welcome.